Welcome back, students. Welcome to Priya's Life Science Academy. I hope you have watched the video nicely and understood everything. So today we will continue with Mendelian genetics. That is very important for UG, PG, for CSIR-NET, Life Science, Get Life Science, ICMR, NEET. Okay, so students pay attention to listen this lecture. Okay. In the previous class, we have already discussed that why we look like our parents and how is this possible. We have got some of the ideas that why we look like our parents because of the genes. And today's class, I want you to know that the discovery, how this discovery came in our subject, that how children look like their parents. How is this possible? The answer is digged by none other than the Gregor Mendel. He studied the gene theory of inheritance. Okay, that is why he is so important to us. We have to learn the Mendelian genetics. Okay, so what he did, what was his discovery? Today we will learn about his discoveries, how he achieved this milestone of genetics. Okay, Mendel used a very important plant, that is the pea plant, Pisum sativum, for his experiment. Okay, now you may wonder that why Mendel used pea plants. These are the pea plants. Why he used them? Because there are some reasons. Whenever you are using a model organism, there will be some reason why you are using it. Like in our daily experiments, we can use mouse, we can use rabbits, we can use C. elegans, everything. But to choose a model organism is very important. That what is your experimental purpose, what you are aiming for, and what this organism or this model plant can give us. So peas are very easy to grow. It's a first important point. They reproduce quickly. That is one very important point to study the generation after generation in genetics. They bears flowers and fruit in the same year that is planted. That is their generation time is less. Produces a large number of seeds. They produce a large number of seeds. That easy to do genetic experiments. So these are the cause why he chose pea plant. Okay. Now, to study pea plants, he have chose 7 characteristic traits in garden pea. So first one is the seed color and the coat color of the flower. Then is the seed color, the seed shape. Then the pod color. Pod is this outer part of the pea. Then is the pod shape. Okay. Then fifth is the stem height, that how much the stem is long. And then is the flower position, where the flower is positioned. So to study these genetic experiments, he used pea plants and he chose these 7 characteristic features. Now next, what he did cross fertilization. I am telling you that these flowers can self-fertilize. They both have these male and female organ inside them. That is why they can self-fertilize. But Mendel did not allow self-fertilize because he wanted to do cross fertilization, the fertilization of a female plant and a male plant, so that in the progeny he could study the genetic experiments nicely. So what he did, he removed the stamen before pollen is produced and retained the pistil and the ovary, and he collect the pollen and he cross fertilized. Okay, and after that the plant seeds, the plants are produced. So what is cross fertilization? It is a simple cross, that is a fusion of male gametes. In this case, the pollen is the male gamete from one individual and the female gametes, that is eggs, from another. As because they can self-fertilize, so Mendel did not allow the self-fertilization. Otherwise, how could we study the genetic experiments? How could we study the parents cross and producing the offspring? So to get the offspring by fertilization, he took the female gamete and crossed it with a male gamete from this is one flower and this is another flower. He did this experiment and he got the progeny. Now this is the Mendel's first experiment. So from the first experiment, this is the parental generation, that is the P generation. This is the purple colored flower. This is the white colored flower. Okay, and it is cross fertilized. I hope you have now understood what is cross fertilization. That is the cross between or fertilization between two different plants. That is not self fertilization, as because plants can, we humans or any other organism, many of the organism we can't self fertilize, but plants can self self-fertilized. To disallow the self-fertilization, he did the cross-fertilization, and I told you that how he did the cross-fertilization experiments. So after the cross-fertilization in the F1 generation, that is the first, F1 means the filial generation, okay. The first generation, in the first filial generation, what he have seen the purple colored flower, okay. By crossing one purple and one white color flower, he got a purple color flower. And then next generation, in the F2 generation, that the filial generation 2, what he got by self-fertilizing them, now these purple flowers are self-fertilized, okay. These purple flowers are self-fertilized, and after self-fertilization, what he could see 3 purple colored flower and 1 white flower, okay, in the ratio 3:1. So what is the observation? So from this, what is proved? So from this, it is proved that Mendel first thought that it is a blended inheritance, that purple and white blended and gave the purple color flower, but his thought of blended inheritance removed when he has seen again the white color flower in the next generation, okay. So from this, he could understand that definitely there are 2 factors responsible for the production of flower colors, okay. That is from P generation, capital P purple and small p white, and when they are fertilized, they are producing capital P, that is purple, okay. Then again, when they are self-fertilized flowers, when they are self-fertilized, it is giving capital P, small p, small p, that is 3 purple and 1 white. So from the first experiment, what is proved? That there is no blended inheritance. There is a thing of dominance, that these 2 factors are responsible, that is the 2 alleles are responsible for flower colors, and one factor is dominant over the other. That is why it is dominant over the small p. That is why we are seeing the purple colored flower. And when in next generation these 2 purple colored flowers are crossed, we are again getting small p, that is the white color flower. He first thought that the small p is vanished, but small p is again expressed when they are coming in dual dose. From here, the law of dominance came into play. In the first lecture, I showed you what is dominant allele, what is recessive allele, but the discovery came from here. In the second generation, this is the F1 generation, this is the F2 generation. In the second generation, he got the white colored flower, and the white colored flower he got because of the presence of 2 recessive alleles. So from here, dominance alleles and recessive alleles thought came into play, and we now know what is dominant allele and what is recessive allele. Okay, I'll show you the cross again very nicely. Look, if this is the parental, this is known as the Punnett square, okay. By the Punnett square method, we can get the offspring very nicely. We have to make a box like this. This is the capital P, this is the small p, and when they cross, for capital P, small p, here capital P, here small p, here small p, here capital P, here capital P, here small p. So you can see all of the offspring are of capital P, small p, that is they are purple in nature because capital P is dominant over small p. Okay, so this is the parental generations cross. Now I will show you the F2 generation, that is, so capital P, small p when crossed with capital P, small p. Okay, first we have to see what type of gametes they can produce. capital P, small p, capital P, small p. Here also capital P, here also small p. In the first generation also, when I showed you this, single type of gamete only, capital P, single type of gamete only, small p. Okay. So before doing the Punnett square, you must know that what type of gametes it can produce. It can produce capital, because gametes are singular in nature. Each copy will be in single dose. I told you, capital P, small p, here capital P, small p. And when we do the cross, here is, suppose these are the two gametes. These are the two gametes. Now the offspring, here capital P, here capital P, small p, here capital P, small p, here small p, small p. So now can you get it? One capital P, two capital P, small p, and one small p, small p. So from here we can get the offsprings, and we can see that 3 are purple and 1 is white. Here we can see how many phenotype? 3:1. 3 is the purple and 1 is white. Okay, and if we see the genotype, what we can see, like capital P, capital P, capital P, small p, capital P, small p, small p, small p. So 1:2:1, because 1 is of capital P, 2 is of capital P, small p, and 1 is of small p, small p. So genotypic ratio is 1:2:1, and phenotypic ratio is 3:1, because 3 purple, but because from outside what we can see that 3 are purple, 1 is white. So phenotype is 3:1 and genotype is 1:2:1. So this is the first Mendelian experiment, and from the first experiment what we can know about the dominance, that because in case of first generation, the capital P, small p, the genotype was capital P, small p. That is why the color of the flower was purple, because capital P is dominant over small p. But in next generation, from the cross, we can get the white flower back because of the presence of small p in double dose. This flower is capital P, small p, capital P, small p, capital P, capital P. From the Punnett square I told you, and I showed you also. So the white color flower is again back when there is a constitution of small p, small p. So from this we got the idea that dominant allele can be produced, dominant phenotype can be produced even in the homozygous condition and even in the heterozygous condition, that capital P is also purple, capital P, small p is also purple. But recessive allele, it will only be expressed when there will be the small p, when only in case of homozygous condition. So from this you can see homozygous capital P, heterozygous capital P, small p, homozygous small p. These are homozygous capital P, purple. This is also capital P, small p, purple. This is small p, that is it. That is why it is white. So we know that genotype refers to an individual genotypic potential, what kind of gene or he or she carries, and phenotype refers to the trait an individual actually shows, the observed condition. Okay. So what is the result of Mendel's experiment from the first cross? So in the F1 generation we did not see any light purple color flowers. In the F1 we only see purple color flowers. There is no light purple color flowers, and because white colors flowers showed up in the F2 generation, in the F2 generation we again can see the white color flowers. So Mendel disproved the blended inheritance. He first thought that there is some blended inheritance of capital P, small p, light purple color flowers or anything, but no, there is pure purple and there is pure white. Purple will be capital P, small p, and white will be small p. Mendel said that the parents had two sets of factors, that is genes, thus two copies of the flower color gene. Yes, from here we could understand that there is two copies of flower color gene, that capital P for purple, or capital P, small p for purple, and small p for white. Okay. There are two sets of factors or two sets of gene, two copies for the flower color gene. That is why this thing is happening. Each gene has two varieties called alleles. You know that there are two varieties called alleles. So one individual can be capital P, one individual can be small p, capital P small p, one individual can be small p, and each gene has two varieties. That is the alleles. Okay, like these individuals has two varieties for its gene, capital P one, another small p. So these two are known as alleles. So from the first experiment, many things came out. The dominant allele, recessive allele, two factors of the gene, and each gene contains two varieties, that is alleles. The concept of alleles also came from the first experiment. In this case, in the case of flower color gene, the two alleles are white and purple. We know that white and purple color genes are present. In the F1 generation, the white allele was hidden by the purple dominant allele. Yes, we know that in the F1 generation, the flowers were purple because the purple color is masked by the dominant allele. So from the dominance, the concept of dominance also came. In the F2 generation, 1/4 of the offspring wound up with two copies of the white allele, thus they were white. Mendel termed this discovery the law of dominance. Okay, so in the F2 generation, what we could see, capital P, capital P small p, and small p. In the F2 generation, white allele came up, and we could see the white allele, and in the F1 generation we couldn't see the white allele. We couldn't see the expression of the white allele because of the presence of the capital P. So this is known as the law of dominance. Because of the dominant allele present, the small allele couldn't be present, but in the F2 generation, two small p alleles were present, so we could get the white allele. So from this, the law of dominance also came into the scene. If only one of the genes in a pair is expressed, it is called the dominant allele. So capital P, small p, if this is the genotype, if capital P's expression can only be seen, that is the purple, then this allele is known as the dominant allele, which we know very before. The gene which is present but not expressed is called the recessive allele. We know from this case. So capital P is present but it is not expressed, so it is known as the recessive allele. So capital P is the dominant one and small p is the recessive one. Okay, recessive alleles may be hidden and not shown for many generations. So yes, we could see that in the F1 generation, recessive allele was hidden, but after the F1 generation, in F2 generation, we could again see the recessive allele, because to express the recessive expression, you need two copies of the recessive allele in order to express itself. When two copies of the recessive allele are present, then the recessive trait will show. Yes, we know in the F2 generation, when when two capital P allele was present, we could see the white phenotype. Yes. Now coming to the Mendel's second law, the law of segregation. Here we have to understand that what is the law of segregation, that the two alleles for each character segregate or separate during gamete formation. I have shown you that when capital P small p was there, these alleles were segregated during gamete formation, capital P small p. Then I showed you through Punnett square that capital P small p, capital P small p, capital P small p, capital P small p. So these two alleles for each character, they segregate or separate during gamete formation. Okay, so Mendel's law of segregation states that the two alleles of a gene that are found on chromosome pair, they separate with the offspring, receiving one from mother and one from father. So according to Mendel's law, the two alleles act in a segregated fashion and do not mix or change each other. They do not mix with each other during gamete formation. They segregate, and the offspring get one allele from father and one allele from mother. So this is the, for example, this is the mother's allele and this is the father's allele. Okay, so this offspring is getting one allele from mother and one allele from father. This I'll show you nicely this thing, like capital P small p, they are segregating, the alleles are segregating during gamete formation. So For example, this is mother and this is father. So this is mother alleles and these are father's alleles. So gametes are produced and offsprings are produced. So this offspring is getting one allele from mother, one allele from father. This offspring also getting one allele from mother, one allele from father. This offspring also getting one allele from mother, one allele from father, and this offspring also getting one allele from mother and one from father. So the thing is that they do not mix with each other. They segregate. The alleles are segregating during gamete formation, and offspring get one allele from mother and one allele from father. So this is known as the law of segregation, that two alleles of each character, they segregate or separate during gamete formation. So this is the second law of Mendel. So first law of dominance, we understood. The second law is the law of segregation. Now we will understand Mendel's third law, that is the law of independent assortment. It is very important. Another thing I forgot to tell you, that this type of cross where Mendel could found the law of dominance and law of segregation, this kind of cross, what I did here, these cross are known as the monohybrid cross. Monohybrid cross means when only one characteristic features is considered. Here the color of the flower is considered, that is purple. Color of the flower is considered, and we could see the genotype for flower color, capital P, capital P, capital P, small p, or small p, small p. So when only one characteristics features is considered, this cross is known as monohybrid cross, because this cross was done between capital P, small p, or between capital P, small p and capital P, small p. So these are known as monohybrid cross. Only one characteristic features is when only one characteristic feature is considered, and mono means one. And what is hybrid? Hybridization means crossing between two parents. Okay, hybridization means crossing between two parents. Very simple. Like, we are not self-crossing. Self-crossing is not hybridization. Hybridization means crossing between two plants or two parents. So this is known as monohybrid cross. And the law of segregation will be between dihybrid cross. So I hope you understood what is di. Di means two. Hybrid means between two parents. And cross means, fertilization. And di means two, means when two features are considered. Like in case of, it could be flower color, seed color, or it could be pod color, pod shape. Okay, it can be anything. So when Mendel crossed peas and looked at two different traits, he discovered that the traits assorted independently. So we are coming to the cross now. Here there are two characteristics features. Here you can see capital Y. Capital Y is for yellow pod color. Small y is for green pod color. Capital R is for round pod shape, and small r is for wrinkled pod shape. So two characteristic features are considered here: color of the pod and the shape of the pod. So this is the color and this is the shape. So here two characteristics features are considered: color of the pod and the shape of the pod. And here you can see that the capital Y is responsible for yellow pod color, small y responsible for green pod color. And from here you can understand that capital Y, that is the yellow pod color, is dominant over small y green pod color, and capital R is for round pod color and it is dominant over small r wrinkled pod color. So this is the scenario. Now when they are crossed, when yellow round, this is yellow and round because capital Y is for yellow, so capital R is for round, is crossed with the green and wrinkled pod, what we got? We got capital Y, small y, capital R, small r genotype. And what will be the phenotype of the plant? What will be the phenotype of the pod? Yes, you guessed right. As because capital Y is dominant over small y, the color of the pod will be yellow. And as because capital R is dominant over small r, the shape of the pod will be round. So the phenotype will be yellow and round. Okay, so here, from here you can see the gametes will be one type only, capital Y, capital R. Gametes will be small y r, and when they are mixed we can get capital Y, small y, we can get capital R, small r. So this is the genotype and the phenotype is yellow and round. And now so this is the F1 generation. Now in F2 generation, when capital Y, small y, capital R, small r, when they are crossed, capital Y, small y, capital R, small r, capital Y, small y, capital R, small r, when they are crossed, we can get these offspring. So from here, what will be the gametes? Here we can get 4 type of gametes from each parent: capital Y, capital R, capital Y, small r, small y, small r, small y, capital R. And from this parent also, capital Y, capital R, capital Y, small r, small y, small r, small y, capital R. So these 4 here, 4 here, 4. So here you can see capital Y, capital R, small y, capital R, capital Y, small r, small y, small r. So 4 gametes from each parent. And when 4 gametes from each parent, when they are producing offspring, you can get this type of phenotype. That is, 9 will be pure round and yellow, 3 will be round and green, 3 will be wrinkled and yellow, 1 will be wrinkled and green. Okay, so this is the expression of the F2 generation, and you can see here by this. So from here, what we could understand, what is the takeaway message from this dihybrid cross? That is, the 2 alleles, like this is the allele for one gene, this is the allele for another gene, like these 2 alleles for 2 or more genes are independent to each other. When they are forming gametes, there is no hard and fast rule that capital Y will go with capital R only, that small y will only go with small r only, or capital Y will only go with small r, or small y will only go with capital R. There is no hard and fast rule. Alleles of 2 or more genes get sorted into gametes independent of each other. They do not depend that capital Y will only go with capital R. You can see here there are lots of type of gametes are produced: capital Y, capital R, capital Y, small r, small y, small r, small y, capital R. So there is no hard and fast rule that only capital Y will only go with capital R or small y will only go with small r. So this is the rule came into play, that is the law of independent assortment, that the alleles of two or more gene will go independent of each other. There is no hard and fast rule that the allele received for one gene does not influence the allele received for other gene. So the alleles are segregated, so the alleles are independently assorted. Okay, so from here the rule came, independent assortment. Alleles are independently assorted during gamete formation. The alleles for one gene does not influence the segregation of the allele for other gene. That capital Y will not influence the segregation of capital R or assortment of capital R. Small y will not influence the assortment of small r, or vice versa. So there can be different type of combination. So they are independently assorted during gamete formation. Okay, that is why we are getting so many offspring. 9:3:3:1. This is the phenotypic ratio. Like we are getting 9 yellow and round, we are getting 3 round and green, we are getting 3 wrinkled and yellow, we are getting 1 wrinkled and green. So the alleles of the 2 or more genes are independently assorted. They do not depend on each other. That is why we are getting so many combination of offspring. Okay, so from here law of independent assortment came into play. So it states that inheritance of one character is always independent of the inheritance of other characters within the same individual. Okay, that is the case. We have seen the yellow color will not influence the shape of the pod, or the shape of the pod characteristics will not influence the color of the pod. So the inheritance of one character is always independent of the inheritance of other characters within the same individuals. Color of the pod will not influence the shape of the pod, or the shape of the pod will not influence the color of the pod. Okay, law of independent assortment is based on dihybrid cross. We have seen that the alleles of 2 or more genes get sorted into gametes independent of each other, that Y will not influence on R, or R will not influence on Y, or small y will not influence R, or small r will not influence, or vice versa, or capital Y will not influence on small r, or small r will not influence on capital Y, or small y will not influence on capital R. Okay, because each of the characteristics are independent of each other. The alleles of 2 or more genes get sorted into gametes independent of each other. The allele received for one gene does not influence the allele received for other gene. Okay, I hope you have understood the 3 basic rules of Mendel. From monohybrid cross we got law of dominance and law of segregation. From dihybrid cross we have understood the law of independent assortment of each character, that one character will not influence other characters. Okay, so I hope you have understood. If you have any queries, you can post it in the mail ID and I'll be happy to give your reply. I'll be back with the next lecture of genetics. I wish you a happy learning, students. All the best for your future. Fight for your dreams and your dreams will fight for you. Thank you, dear students.